Good morning. It's July 10th, and this is your daily brief in science. Here's everything you need to know. Researchers analyzing data from over 5,000 children with autism have identified 4 distinct subtypes of autism spectrum disorder, each with unique genetic profiles and developmental patterns. These subtypes are social and behavioral challenges, mixed ASD with developmental delay, moderate challenges, and broadly affected. The broadly affected group, comprising about 10% of cases, exhibits the most severe symptoms, including profound delays and multiple genetic mutations, and faces higher risks of additional psychiatric conditions. Children in the mixed ASD with developmental delay group, around 19%, tend to lag in milestones like speech and walking, with more variability in core traits and a higher likelihood of carrying rare inherited genetic variants. The moderate challenges group shows milder core symptoms and more typical development, while the social and behavioral challenges group is linked to genes involved in brain development and is diagnosed later. Genetic analysis reveals that each subtype is associated with specific mechanisms. For example, the broadly affected group has the highest number of de novo mutations, whereas the mixed ASD with developmental delay group more often carries inherited variants. Different biological pathways are affected across subtypes, such as chromatin organization and DNA repair in the social and behavioral challenges group, and neuronal signaling issues in the mixed ASD with developmental delay group. Current genetic testing explains only about 20% of autism cases, with environmental factors, DNA modifications, and gene-environment interactions accounting for the remaining 80%. There is concern about potential overdiagnosis in England due to inconsistent screening practices and diagnostic criteria amid rising autism diagnoses globally. Overall, autism diagnoses have increased significantly, with estimates of 2.3 million children and 7 million adults affected in the U.S., driven by better screening and awareness. This research supports ongoing efforts to investigate environmental influences on autism rates and provides a framework for understanding other complex neurodevelopmental conditions. A new study from the University of Pittsburgh shows that exercise can improve cancer treatment outcomes in mice by changing their gut microbiome. Specifically, exercise increases levels of a metabolite called formate, which enhances the immune response against tumors. Formate boosts the activity of CD8 T cells, which are crucial for destroying cancer cells. When mice were given oral formate, tumor growth was significantly inhibited, and survival rates improved across different cancer models. Mice that underwent exercise had smaller tumors and better survival in melanoma studies, but these benefits disappeared when their microbiome was eliminated with antibiotics. This indicates the microbiome plays a vital role in mediating exercise's effects on cancer. The research was funded by multiple organizations, including the NIH, the Melanoma Research Alliance, and the Burroughs Wellcome Fund. Scientists identified microbiota-derived formate as the key compound through machine learning analysis of metabolic pathways. Experts highlight the importance of studying bacterial metabolites rather than just microbiome composition, emphasizing the need for further research on the persistence of exercise benefits. The findings suggest a shift from focusing solely on bacterial species to understanding the metabolites they produce, which could help improve immunotherapy. Future studies aim to explore how exercise influences the microbiome in other diseases like autoimmune disorders. In human melanoma patients, higher blood levels of formate were linked to better progression-free survival during immunotherapy, suggesting a potential connection between exercise, microbiome metabolites, and cancer outcomes. Scientists have developed a new artificial skin made from a flexible, conductive, gelatin-based material that mimics many properties of human skin. This innovative material can be shaped into various forms and embedded with electrodes to sense touch, temperature, pain, and environmental changes. It responds to stimuli by conducting electricity, expanding, or releasing substances, similar to biological responses like sweating. The skin is self-healing within seconds and combines graphene with conductive polymers, making it durable and capable of sensing multiple types of stimuli with a single sensor. This reduces complexity and cost compared to traditional electronic skins that require multiple sensors. The research, published in Science Robotics on June 11th, aims to enhance robotics, prosthetics, and military applications, including creating self-healing robots and miniature vehicles capable of detecting and disabling threats. Over 860,000 data points from tests on robotic hands were used to train machine learning models to recognize different types of touch. The skin can detect signals like temperature, pH, pressure, humidity, and biological markers, responding by changing conductivity or releasing substances. While not yet matching human skin's full capabilities, it outperforms existing options and can be calibrated with human touch. The development marks the first time a material with properties identical to natural tissue has been created, opening new possibilities for biomedical and technological advancements. Rising ocean temperatures in the Arctic are linked to an increase in harmful algal blooms that produce toxins affecting marine life and human health. Over the past 20 years, analysis of whale feces has shown a clear connection between warmer waters and higher levels of toxins like domoic acid and saxitoxin entering the marine food chain. Environmental data indicates that the loss of sea ice and specific wind patterns promote these blooms by allowing more sunlight to warm the ocean surface. As sea ice diminishes, more sunlight penetrates, fueling algal growth that is now more prevalent and toxic than in previous decades. Higher sea surface temperatures in July and increased open water in June are associated with elevated toxin levels in autumn, suggesting earlier and more widespread harmful algal bloom conditions. Warming waters accelerate the germination of toxic algae cysts on the seafloor, especially in the sediments of the Alaskan Arctic, increasing bloom frequency and toxicity. The proliferation of species like Pseudonitschia and Alexandrium catanella, which produce domoic acid and saxitoxin respectively, is a direct consequence of these changing conditions. Monitoring harmful algal blooms has become essential as toxin levels and bloom occurrences continue to rise, threatening marine ecosystems and food security for indigenous Arctic communities. These communities are now seeking tools such as field tests to monitor toxin levels in traditional foods, as neurotoxins increasingly impact marine mammals and humans. Environmental factors like stronger northeasterly winds and the presence of the Beaufort high-pressure system are associated with lower toxin levels by reducing heat flux, while weaker winds and reduced pressure correlate with higher toxin prevalence. Interdisciplinary collaboration among scientists, indigenous knowledge holders, and government agencies underscores the importance of teamwork in addressing these challenges. The increasing frequency and toxicity of algal blooms highlight the urgent need for ongoing research and adaptive management strategies to safeguard Arctic ecosystems and communities. Researchers at Stanford Medicine have developed a blood-based algorithm that assesses the biological age of 11 organs in individuals aged 40 to 70. This method analyzes nearly 3,000 proteins from blood samples of over 44,000 UK biobank participants collected over 17 years. The approach reveals that different organs age at varying rates and can predict future disease risks and lifespan. It marks a shift from reactive to proactive healthcare, focusing on early detection of aging-related health issues before symptoms appear. A youthful brain is associated with a 60% reduction in mortality risk, while a healthy immune system correlates with increased longevity. Conversely, a biologically aged brain significantly raises the risk of Alzheimer's disease, with an extremely aged brain increasing the likelihood of developing the disease over 10 years by about 12 times. Brain age is the strongest predictor of overall mortality, with an extremely aged brain increasing death risk by 182%, and a youthful brain reducing it by 40%. Stanford researchers plan to commercialize this organ-age testing technology within 2 to 3 years through licensing, initially focusing on the brain, heart, and immune system. The goal is to develop this research into consumer health products that enable early interventions and personalized health strategies. Funding for the study was provided by organizations including the NIH, the Milky Way Foundation, and the Stanford Alzheimer's Disease Research Center. This organ-specific biological age assessment could transform preventive medicine by allowing early intervention before disease symptoms manifest, especially for conditions like Alzheimer's. About 1/3 of participants showed at least one organ with accelerated or decelerated aging, with the brain showing the strongest link to health outcomes. The algorithm uses machine learning to compare protein signatures against population averages, classifying organs as extremely aged or extremely youthful based on deviations from the norm. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.